Hello to all UTHIS participants. I'm really proud of being here to show you what is the immune system and how the immune system works. Let's have a look to different parts of the components of this system, was of the most complex in nature, and including molecules, cells, tissues, and complete parts of our body, all perfectly connected. So let me start with telling you that the immune response acts in 3 levels of complexity if we attend to from the outside of our body to the very good inside. So first part, all our body is protected with a kind of wall, which is our skin, and all the epithelial tissues which are covering our eyes, our respiratory conducts, our gastrointestinal conduct, our genital, and also our urinary conducts. All of them are covered by epithelial cells. It's like a thing similar at a wall, like you see over here, with some cells doing the physical separation between the inside and the outside of our body, and some other cells secreting different substances to make it difficult for the microbes to grow over there. So we have a physical barrier, a chemical barrier, but not only, we have also a biological barrier. Let me show you how this is really. This barrier is not like this, but like that. So this wall is covered of thousands of different microbes, mainly of them bacterials, growing calmly over there and helping each other. So the bacterials help us to defend our body of pathogens, and our body also helps bacteria to grow. So it's a commensal structure and it doesn't hurt. So first level of our immune system, don't forget never, our skin and our mucosal tissues. What else? If this is good, but it's not enough to defend after an aggression. And for example, think if you're brushing your teeth very strongly and you make hard on your mouth. Or think on summer you're walking barefoot over the seaside and something hurts your feet. So what happens? This wall is broken. What is the next step in our immune defenses? Is what we call natural immunity or inspecific immunity. There's a collection of cells and molecules, we call it natural immunity, that doesn't have any memory of what's going on. They could kill the bacteria, the virus, the fungus that is infecting us. Here are things like we call complement system, for example, or defensins, or any microbial peptide, which is also in our saliva or in our tears. We have all kind of endogenic antibiotics to kill any microbe. So this is natural defenses, natural immune response, and doesn't have any memory. What it means, if today I have an infection, and in 3 years' time I have the same infection, these natural cells doesn't remember this pathogen and are acting exactly the same. And this natural immune response also secretes some substances giving information to the third and most sophisticated step in our defenses. These substances giving information to these special cells are called cytokines. And there's a lot of different cytokines, kind of immune hormones, and run from one cell to other, giving a message, giving special information to have any given action. So what's the end of the natural immune response? This second step. We have some cells, phagocytes, killer cells, complement system, defensins, and also cytokines acting against the infections, against the pathogen, but it's not enough. We need the third level, the sophisticated step of our defenses, is what we call the adaptive or a specific immune system. And it's the part we are speaking much more today. So anything starts when a pathogen infects a cell. Let's have a look how it occurs. Here is a pathogen. I'm selecting the SARS-CoV-2 because it's quite common these days. And you see there's a lot of different proteins over the structure of this virus. But let's have a look especially to this red one. The red one is a protein called the spike, and it's a kind of key to enter in our cells, because in our cells we have some kind of receptor which adapts perfectly to this protein. So the protein interacts with the receptor and open perfectly our cells, and the virus could enter inside. What's next? Next, this cell has to say the specific immune system that here is an infection. How this occurs? This virus is completely digested inside the cells in parts. You see the green parts, the blue parts, yellow parts, and also red parts of this virus. Remember, 4 colors as an example. And what's happening with these parts of the virus is show outside the cell, inside the specific molecules we have in all our cells. These are called antigen-presenting molecules because it presents bites of this antigen. You see this yellow one doesn't adapt. We have another one here. This blue one is too big to this one, but it's perfect for this other piece. This yellow, it's perfect here. This blue is perfect here, for example. So the cells show to the immune system there is an infection, and you have parts of the virus in this case saying, I have a virus inside. Who is looking at these facts? Who is looking to this infection? Was the first cell to interact is what we call the T helper cell. It's the most important cell in our defenses and has a special protein in the surface to interact with these molecules. So this is the important cell. Have a look, say what's going on, and have to decide what to do. And the specific immune response has different ways to answer this infection, but we are resuming this in two ways to answer this infection. So this man, the T helper cell, I remind you, look at the infection and say, well, I could clear this infection killing these cells. So if we eliminate the infected cells, it's a way to stop the infection, doesn't it? Yes, was how this immune system kills this infected cell. I have to turn the cell to show you better. How this boy killed this cell. But he doesn't kill directly, he needs another cell. A second cell is a second T cell, it's a cytotoxic T cell, it's a very dangerous cell, but it's not specifically trained, it's a bit chaotic itself, but kills this infected cell. How? Shooting a collection of enzymes that directly induce the dead cell. We call this process apoptosis. It's a kind of cell suicide. So the cell's death itself. This is the first way, but it's not the only. I have told you there's a resume of 2 ways of helping our body to defense. The second way is to generate our best weapon in nature to kill or to block an infection, which is the very well-known antibodies. But how an antibody is born? So this boy now needs a third party, which is the B cell. The B cells is another specific immune cell that specifically has a protein in its surface that specifically interact with a given antigen of a given pathogen. This the antibody, could be in the cell surface, as you see here, but is also secreted. So the antibodies has two parts interacting with the antigen. Here we have an example of an antibody with two parts interacting with the antigen. So in a given antigen, the red one recognizes perfectly and very specifically only the red protein. So in this infection, we would have at the end of the day red antigens for the spike, but also blue antigens for the blue protein, also yellow antibodies for the yellow protein, and finally green antibodies for the green protein. So our immune system is secreting all of these antibodies in our blood. And these are called, or the best preferred antibodies are the so-called neutralizing antibodies. What does it mean? It means a very simple thing. If an antibody recognizes specifically the red protein, blocks like this way, as you see in here, and now this virus can't interact with the receptor on our cells. So this virus can't infect anymore. It's blocked, and we call this neutralizing antibodies. So the best way to be immunized nowadays against SARS-CoV-2 is to having in our blood neutralizing antibodies against the red, which is the spike protein. This is the only way to obtain the antibodies? No, because this way is generating memory in our blood. We are also using vaccination. This is the natural infection, but we could do also vaccination. For this, I will show you in a minute a big virus structure. It has a surface of virus. A virus is not alive. It's not a living thing because it needs the cell itself to grow inside. So you see the proteins, and in the inside, the virus has the molecular information to build the virus itself. So the molecular information is an RNA, in this case, with the 4 colors. The information to build the red one, to build the blue one, the yellow one, and finally the green one. So how the vaccine works? In the lab, in the laboratories, scientists have isolated the red part, which is the one coding for the red protein, the one called spike, the one entering the cells. So the red protein. Could we vaccinate just with the red RNA? No. This is a very unstable molecule, so it needs to be encapsulated and stabilized. This had been done with nanocapsules, nanoparticles, which are more or less different kinds of lipids that capsulate this RNA. So these are the so-called RNA vaccines, like Pfizer and Moderna brands that are running these days in our countries. RNA vaccines. These lipids in the outside are exactly the same as our cell surface, so it could enter our cells without any problem. So this was the first vaccine in the world for SARS-CoV-2. And what was the second ones? The second ones were based on adenovirus. Adenovirus is just a vector. It's a virus we have very common in the colds, on the winter, and we have it also infecting our eyes. We call that conjunctivitis, adenoviral conjunctivitis. So it's a different family of virus. And what scientists have done for these vaccines is open the virus and modifying the DNA inside. The DNA has 2 parts. The white part is the envelope of the virus itself, and the dark one is the infectious parts of the virus, so they have split it. and they have insert here the red part of our spike, our coronavirus. Again, this is placed inside our virus, and now they have infected the cells with this. And yes, that the question you are asking yourself is yes, in our cells we have a receptor for this adenovirus, so it could open our cells exactly the same and inject the RNA, the DNA in this case, inside our cells and producing the immune response. As you could see, with both adenovirus vaccines and also with RNA vaccines, I don't know where is it now, inside, sorry. We are only stimulating the red part of our immune system. So at the end of the day, we are only having the red antibodies, and these red antibodies are blocking specifically the spike protein and blocking specifically the infection of this virus inside our cells. And this is how this vaccine works. We don't know at this moment if these antibodies are lasting for very long, but scientists are doing this job, and we will see in the next future. I hope now you look more after your immune system and take care of different things you could do to get your defenses up to the top. Thank you very much, and see you soon.